Hi friends, how are you? Okay, you know that we have been studying about the chapter in science, that is life process. We have already studied about the life process. What is life process and what are the importance of life process in living organisms and what are life process. All these things we have studied already, and the first life process, that is nutrition, we are studying now. In nutrition we have studied the variety of organisms which are having a mode of nutrition, variety mode of nutrition, autotrophic, heterotrophic. Like that, in heterotrophic we have studied three types of nutritions, and in that we are now studying the autotrophic nutrients, how they are preparing their own food. In this concept already we have studied about photosynthesis. What is photosynthesis and all these things, how they are preparing themselves food, that is green plants, how they are preparing themselves their food and how they are storing their foods. All these things we have studied in last video. Now we are going to study in photosynthesis. There we have seen that three important minerals are there for photosynthesis. So for photosynthesis, three are necessary. Those three are: first one, sunlight. We have studied when about photosynthesis, when we are studying about that, we have seen there sunlight should be there. Because of that only plants are going to photosynthesis at the daytime. So sunlight is necessary for photosynthesis. We have studied that. And next one is, friends, carbon dioxide, which is in the environment, that carbon dioxide. And we have studied that carbon dioxide. And next, water. Carbon dioxide and waters are inorganic materials which are in the environment. By using that inorganic minerals, plants are going to be convert them into organic minerals or chemical energy. They are transferring. So these three minerals are necessary. And the next one is chlorophyll. So this is also very important in photosynthesis, which is present in the plant cell itself. So when all these minerals are there, then only photosynthesis will be taking place. So these are the basic things we have studied in our last video. Now on the basis of these things, we are going to make an experiment to show that sunlight is necessary for photosynthesis. We have said that these four are very necessary for the photosynthesis, and we are going to make an experiment to show that how these four are necessary for photosynthesis. If in these four, if anyone is not there, what will be happened? That we are going to experiment in this video. In that, first, if the sunlight is not there, what will be happen, or why and how sunlight is necessary for the photosynthesis. This concept we are going to study in this video, friends. Okay, if we goes to sunlight, in last video I have told you, when the sunlight comes to the leaf, when it fell on the leaf, the leaf will be reflect green and yellow color, and other two color it will be absorbed. When green and yellow color will be reflect, we will see the color of leaf is yellow mix green, because only two colors this is reflecting, and all the other two colors it is absorbing. That I have told in our last video. But the question arises here. We have seen that in our rainbow 7 colors are there, but here we are telling that 4 colors are coming to the leaf. Then how it is? So I have to explain that point today in this video. See, if you see the rays of the sunlight, it is having like this, the atoms are coming out of the sun like this path, and this path is having various enormous rays. Those rays are dividing in the base of the wavelength and frequency of the light, and wavelength and frequency are very important. So in these rays, they are divided by the wavelength of the light and frequency. The frequency and wavelength are inversely proportional each other. If the wavelength is big, frequency is small. I will explain it in our next video, that is in the light in topic, that I will explain in detail about this. But here we are going to take the variety of rays in sunlight. That is gamma rays are there, and X rays are there, UV rays are there, and infrared rays are there, and radio waves are there in sunlight. But in these four, between there is a visible light part is there. In visible light, that we are saying that the 7 colors of the rainbow are there in visible light of the sunlight, because the wavelength of these rays are like that much they are bigger, that we are able to see those lights by belonging their wavelength. That wavelength we can see in our eyes. So because of that, this part of the rays we are calling it as this calls visible light. And this visible light we are seeing here, when the rays goes on, the wavelength is going to be increasing. So the first one is violet, or last is red. So violet is the first color coming in the sun rays, which is having a very short wavelength compared to the red. So because of this short wavelength, the space occupying is very small. Like that, the wavelength goes on becoming broad, the light length will be goes on increasing. Like that, blue color, before blue color, all rays are having very small portion in the rays because of wavelength. So because naturally, if you see that the blue color is having the same all the part of this, because all the colors before blue color are merged into the blue color. So we are feeling that only 4 colors are there, that in the visible light. So because of that, we have told that 4 colors are there and 2 colors are reflecting and other 2 colors are emerging. So 2 colors means in the blue, all other colors are emerging, mixing. See here how they are mixing. In totally, generally it is feeling that it is blue color. So like that, 4 colors means 7 colors are there which are landing on the leaf. And these 7 colors, 2 colors are reflecting from the leaves. Like that. That is yellow color and green color. These 2 are reflecting from the See, yellow color and green is reflecting from the leaf. So because of that, this reflection, we feel that leaf is yellow mixed green color. So okay, if this is reflecting and other 5 colors are emerging in the leaf. This basic point you have to bear in mind, because this point is important when I am explaining the experiment of sunlight is necessary. In that experiment, this point should be arise there. So as a basic point, I am explaining it. Other 5 colors are emerging on the leaf. These 5 colors are going to the chlorophyll, up to chlorophyll. Okay, when these colors are land on the chlorophyll chlorophyll become active and the photosynthesis will be start. This is the basic point of photosynthesis. Okay, next you come the point. Let me explain the experiment outline to explain this point. See there, friends, what our aim is today. We have to prove sunlight is necessary for the photosynthesis. What it means? If sunlight is not there, photosynthesis mechanism will not going to be made by the plant. This point we have to explain. Means sunlight is necessary for the photosynthesis, that we have to prove here. Okay, for that, see friends, if sunlight in leaf we are explaining the chemical reaction. We are proving this point in the experiment by the chemical reaction, that is when iodine is mixed with the starch, the iodine will be converted in the color blue-black color. This chemical reaction we are using in this experiment. This is the important step. Finally, we are taking this result. Okay. So, where the starch is there in leaf? It comes in leaf by photosynthesis. When the photosynthesis happen, then glucose will be formed. That glucose will be converted into starch. It will be stored in the leaf. So that starch will there in the leaf because of photosynthesis. If photosynthesis is not there, then starch will be not ready. Starch is not there, then that portion where starch is not there, when you put the iodine on that part, it will not going to be converted into blue-black color. This is the outline. Okay. Starch is necessary for our experiment. This is I have explained. So when you take a leaf, we know that in the leaf already there is a starch. It is stored. So to remove the starch, we are going to put it in dark place, and after that we are going to make this leaf covered by a paper, and this covered paper, this portion will not allow the sunlight. Because of that, photosynthesis will not going to be taking place here. Then we are going to wash it with alcohol. I will explain it how and why. And last, we are taking this leaf and putting iodine. It will be converted into blue-black color. I will explain it. I am telling the outline of the experiment. Then the question arises here, why iodine is converting the starch into blue-black color? This is the question, this is the basic question. And the chemistry is here only. That point as a basic thing I am going to be explained here in brief, not in detail. But see friends, if you have to understand any concept, it will be understood by you people when there is no question in your mind about that concept. About that concept, no confusion will be there, then only you will be clear about the concept. So that part, I want to clear you this concept. Because of that, I am explaining the basic things about the concept. Okay. So when starch and iodine will be mixed together, what will be happen? Iodine is having 3 iodide ions, means in straight way these ions are handling in straight way. So this is the structure of the iodine. 3 iodide ions are in straight way in iodine. And comes to the starch, it is having 2 amylase and amylopectin, 2 chemicals it is having in the starch. And amylase is a straight structure. It is having a straight structure and helix type of structure, means this type of structure we are calling it as helix shape. And if you come to the amylase pectin, it is very complicated helix structure because it is having branch shape. And these 2 are in the starch. Because of that, the starch is a very complicated structure that I have told you. It is very difficult to digest. Because of this structure, starch is very complex food. So this type of complex structure it is having. If you mixed iodine and starch, this iodine ions, which are 3 in number, these are going to be correctly mixed with the complicated structure of starch like this. See here, how they are correctly structured in the starch structure. So this is the mixing of iodine and starch. When this will be happen, there will be prepared I3, a single electron complex. Starch 3 iodide complex. When this complex will be happen, the nature of this complex is attracting all the color which are fell in that complex. This is the main scientific reason here. The basic thing. When all the colors are absorbing by this complex, it means what will happen? I have explained in first point. 2 colors are reflecting from the leaf and other all 5 colors are absorbing by the leaf. So it means when the sunlight comes to the leaf, comes to the chlorophyll, 5 type colors are there in the sunlight. So those 5 colors are coming to the mixture, and that mixture is absorbing all the 5 colors. So you can try, friends, without green and yellow. You mix all the 5 colors. You will be see there that we have to mix in the ratio what the wavelength is there. Means violet is very small portion, blue is heavy portion, red is very heavy portion. Like that, if you goes on mixing 5 colors, you will get blue-black mixed color. So that is the reason when you mix iodine with the starch, you will get blue-black color. This is the basic concept. Okay, friends. And second one is, if you mix iodine with green color, means with the leaf in green color. Okay, I will explain it when we are making experiment. Okay, these 2, one the color, rays color, and second one is mixing of iodine and starch as the basic thing. If you come to the experiment, now we are going to start the experiment. Okay, friends, when we are going to start the experiment, we need some apparatus. Those are, first one is potted plant. What is potted plant? In a pot, we have to we have a green plant. That plant should be on the pot. That pot we are calling it as potted plant, which is in the pot. Next one, thin aluminum paper. Next one, petri dish. Next, beaker, alcohol, water, iodine, burner and dropper. These 9 things we need in the experiment. Okay, come to end. In the outline of the experiment, we have said that starch is there. If starch is there, it means what happened? Photosynthesis is happening there. Then only the starch will come. If we show that the starch is there in the leaf, then by that we are going to be proved that photosynthesis is happening here. Okay, so starch is there. By the basis of starch we are proving this experiment, means we have to prepare the starch after when we are starting the experiment, then only we have to prepare that starch. If the previous starch is there, then it will not give the correct reading at the end of the experiment. So first, when we are starting the experiment on that plant, there should be no balance of the starch which is prepared by those and stocked there in the plant. That we have to remove completely from the plant. Means it will be de-starched. The plant should be de-starched before we are starting the experiment. That we have to make first. So for that we are keeping the plant, potted plant, in the dark place for 3 days. What will happen by this? We are keeping it in a dark place, means we are avoiding the sunlight from the plant. When sunlight is not there, then photosynthesis should be not happening there. Mechanism is not going to be made by the plant. So photosynthesis is not there, glucose is not forming. So glucose is not there, starch is not going to be stored. So because of that, the stored starch will be used by the plant for their food. Glucose is not preparing. So what will be done by the plant? It will use all the stored starch to alive. For 3 days, what the stored starch is there, complete starch will be used by the plant. After the end of 3 days, all the starch will be used by the plant and the whole plant become de-starch. No starch. The stock percentage of starch is 0 in that plant. That plant we have to use in our experiment. For that reason, we are keeping the potted plant in the dark place for 3 days. That potted plant is we are now using in this experiment. So this is the plant which is kept 3 days in the sunlight. In the dark place. Next, what we are doing? We are covering some portion. Partly we are covering one leaf by the aluminum or black paper. This is because now we are going to be kept it in a sunlight. When we are keeping it in a sunlight, when the sunlight fell on the leaves, again the plant started the mechanism, photosynthesis will be start. Photosynthesis will be start in the plant. So photosynthesis is going to be start, then the glucose and starch will be going to stored. So to avoid this portion, sunlight is not there. If the sunlight is not there for this portion, then what we are saying? For photosynthesis, sunlight is necessary. So if sunlight is not there, then according to our statement, there should not be a photosynthesis. because sunlight is not there. What we are saying? Sunlight is necessary for photosynthesis. That is our statement. Now we have to prove that. So for that, what we are doing? We have to prove that. So because of that, we are covering partially the leaf. Means we are avoiding the sunlight from this portion. So according to our statement, if sunlight is not there in those portion, there is no going to be photosynthesis. So because of that, we have to cover the leaf by the aluminum paper or black paper, and it should be tight to the leaf. Because of that, we have to keep the paper clip on that the rays not enter to the portion by sideway. We have to avoid that. We are keeping clips on the paper clips on the leaf. Another one method is also there. There is a stand. We are calling it as Gang's light stand. What will happen? It will clip the leaf. It will clip the leaf like that. If it is a leaf, it is going to be clipped like this. In this portion, sunlight is not covering, not falling on the portion of that clip. So maybe if in every experiment we are unable to adjust this stand, so because of that we are using the aluminum or black papers here. Okay, next. See here. We are keeping that partially covered plant in a sunlight, heavy, bright sunlight. So when you kept the plant in bright sunlight, what will happen? All the portions of the plant will start photosynthesis. According to our statement, in that portion which we have covered, there should not be a photosynthesis because sunlight is not there. If sunlight is necessary for photosynthesis, then in that portion photosynthesis will not going to be taking place. We will check it afterwards. So to check it, we are making cover that portion and we are keeping it in the sunlight, bright sunlight, and for 3 days we are keeping it. Because of this period, the glucose will be formed and that glucose will be going to convert into starch. So all the portions, again, what the plant has become de-starch, now it becomes stored the starch. If the statement is correct, in that covered portion, starch is not going to be stored. We will check it. Okay. Now we are taking that leaf from the plant. We are separating that leaf from the plant. And we are keeping that leaf in a boiled water. We are going to keep it in a boiled water. This is because the outer membrane of the leaf will going to be damaged by this. Will be going to be break by this. By breaking this outer layer, outer membrane, we are able to see the inner portion of the leaf clearly. Otherwise it will be confused. The result is not clear, maybe. So to avoid that, we have to break the outer membrane of the leaf. Because of that, we are keeping it in a boiling water. That boiling water is damaging or breaking the outer layer of the cells. Okay. For that, we are keeping it in a boiling water. Next, we are keeping it in a alcohol, boiling alcohol. This is why, isn't it? That here we are keeping it in a alcohol, that is a boiling alcohol. This is because, see friends, the leaf is having chlorophyll. We are not disturbing the chlorophyll by covering the paper. Chlorophyll is there in the leaf. We are only disturbing the starch, sunlight. We are disturbing the sunlight. By that, we are thinking that starch is disturbed. Okay, sunlight we are disturbing here, but not chlorophyll. in the leaf, in this portion also there is a chlorophyll. And we know that if the chlorophyll comes to contact with iodine, that result is also as same as the iodine and starch result. Means if the green portion of the color, that is chlorophyll, mixed with the iodine, it is also going to be convert the iodine into blue-black color. So to avoid that confusion result, we have to remove completely chlorophyll in this portion. So to remove the chlorophyll, we are keeping it in a boiling alcohol. By this, what will be happen? That boiling alcohol absorbs the chlorophyll and alcohol become green and leaf become white. All the chlorophyll it will absorbing from the leaf. Okay, then if the alcohol, boiling alcohol, we have to keep, but another one beaker is there. Why we are keeping two beakers here? So see friends, we know that alcohol is heavy, attractive by the fire. We know that it will be get fire immediately, just like a petrol. Alcohol is also inflammable, heavy inflammable liquid. So if you keep it with directly contact with the fire, it will take the fire. It will immediately take the fire. To avoid that portion, we are keeping the alcohol in a water filled beaker. When the water is going to be start in boiling, that boiling water is going to be make heat to the alcohol. Alcohol is going to be boil by this water. We are avoiding the direct contact of alcohol and fire. To avoid that, we are keeping the alcohol beaker in a water filled beaker. When the water is boiling, by the help of that heat, alcohol is going to be boiled. When the alcohol is boiling, we are keeping the leaf which is having chlorophyll in this. We will keep the leaf in the boiling alcohol. By this, what will be happen? Yes, all the chlorophyll will be absorbed by the boiling alcohol and the leaf become white and the alcohol become green. So by that, the paled leaf which is white in color, we are removing from the alcohol, and we are tapping with hot water the leaf. With hot water, we are washing the leaf. This is because when we are kept it in a boiling alcohol, all the chlorophyll will be absorbed by the alcohol. Even also, this leaf is in the alcohol only, which is having absorbed by the alcohol, which is absorbed chlorophyll. In that alcohol, it is present. By this, what will happen? Some portion of the chlorophyll will be attached to the leaf. If that is there, it is also disturbing our correct result. So to avoid this confusion, again, to get the correct result, we have to remove all the portions of the chlorophyll which is in contact with the leaf. To make it, we are washing it in a hot water. By this, all the chlorophyll which is there in contact with the leaf will be removed by the leaf. Means now it is completely clear by the chlorophyll. Pale leaf, white leaf, now it is. And next, this white leaf is kept in a petri dish, which is a small dish like this. We are keeping it on the Petri dish, and with the help of the dropper, we are pouring iodine on that leaf. So see friends, this is a leaf which is completely clear by the chlorophyll. Chlorophyll is not there in this leaf. So chlorophyll and iodine reaction is not able to, not will be going to become here because chlorophyll is not there. Okay. Next, in the covered portion, there is no sunlight. Because of that, according to our imagine, starch will be not there. Because sunlight is not there, if the sunlight is necessary for the photosynthesis, then the photosynthesis is not going to be taking place on that covered portion and starch is not prepared. So, according to us, here in the covered portion, there is no starch. But uncovered portions are there which is having starch because the sunlight is fell on this uncovered portion and chlorophyll was there. So this chlorophyll prepared the starch by photosynthesis. So what will be our imagine, in the non-covered portion there should be a starch, in covered portion there is no starch. This is our thinking. Let it is right or not in the experiment. When we pouring the iodine on the leaf, what will happen? Yes friends, where starch is there, in what portion starch is there, that portion should be converted into blue-black color because starch and iodine are mixing there. And if this covered portion is not having any starch, then iodine is not affected in this part. Means the color is not going to be changed in this covered part because the reaction is not going to be there. Starch is not there, then the reaction is not there. So our result should be that in covered portion color is not changing, the leaf should be in white color. In uncovered portion it should be become a blue-black color. Okay friends, let us check the result. See friends, this is the stand we are using here. Stand used leaf. In stand there is a hole like this in the stand and other all portions are covered by the stand. So in this portion sunlight is there entering in this portion in that stand. So in other all portion sunlight is not entering there. So where the sunlight is not entered, in this portion the color is not going to be changed. Means it becomes brown color because of iodine. The iodine natural color, it is brown. But the other portion become blue-black color. Where the blue-black color is there, it means it is absorbing all the 5 color. When it will be absorbed 5 color? When the starch and iodine are taking part in the chemical reaction. So it means it is taking blue-black, means here the iodine is taking part in the chemical reaction. Means there is a starch is there. In this portion starch is there. So this portion is that portion which the sun rays are covered because this is uncovered portion. So because of that photosynthesis has happened and starch will be prepared and the color will be changed. In the covered portion there is no starch. Because of that only it has become only brown, not blue-black. So when it is brown, it means what? There is no starch. Then it is proved that when sunlight is not there, starch is not there. Starch is not there means photosynthesis is not there. So what the conclusion is? If the sunlight is not there, photosynthesis will not going to be taking place. Means sunlight is necessary for the photosynthesis. This result it is proving. You understand, friends? The basic thing, the basic science behind this concept, behind this experiment, you have to understand. If you understand the whole concept technically, then only you are able to understand it and you are able to remember it. For that reason, I have explained every scientific points behind this concept. So we will take a revision of this experiment in short. Okay, friends. First, what we have done? We have kept the potted plant for 3 days in dark room because to de-starch the plant, whole plant. The de-starched plant is covered. In that de-starched plant, one leaf is partly covered by aluminum or black or by the stand, black paper or by the stand. By that we are avoiding sunlight from that portion, and that partly covered leaf with plant with leaf, we kept it in a bright sunlight. By that, what we have done? We have allowed the plant to contact with sunlight and start the photosynthesis. And next, we are taking that leaf in a boiling water. By this, what will happen? All the membranes of the cells will be damaged. By this, we can see the result clearly. Next, we are keeping it in the ethanol or alcohol. In the boiling alcohol, by that alcohol mixed with the green portion of the leaf, and the leaf becomes pale white color. Chlorophyll will be removed by the leaf. That chlorophyll less leaf we are going to keep on the petri dish, and we are pouring the iodine solution on that leaf. By this, where the photosynthesis is taking place, in that portion iodine become blue black. Where the photosynthesis is not taking place, means the covered portion is not having starch. Because of that, that will be remains in the brown color only. By this, we have proved that where the sunlight is not there, there the photosynthesis is not happening. Because of that, starch is not storing, and starch is not there. By that only, the iodine is not affected on that portion. This is our result, friends. You understand? If you are any doubts in this experiment, please comment on the video and like it and share it. Okay, friends, we will meet again in the next video. Bye.